Hi guys, welcome to my video, and in this video we will discuss the protein synthesis. This amazing artwork shows a process that takes place in the cells of all living organisms and all living things in the world. The production of proteins. This process is called protein synthesis. It is actually consists of two processes: the transcription, in which we transcribe, we rewrite in this process, and translation, which we translate, recodes the RNA, and we will discuss it further. In eukaryotic cells, transcription takes place in the nucleus. During transcription, DNA is used as a template to make a molecule of messenger RNA. mRNA, also known as messenger RNA, can is transcribed and written the information as new mRNA. The molecule of messenger RNA then leaves the nucleus and goes to ribosomes in the cytoplasm, where translation occurs. During translation, the genetic code in mRNA is read and used to make a protein. These two processes are summed up by the central dogma of molecular biology, from DNA to RNA is the transcription, and from RNA to protein is known as translation. So first, in transcription is the first part of the central dogma of molecular biology, where DNA is converted to RNA. It is transfer of genetic instructions in DNA to mRNA. During transcription, a strand of mRNA is made to complement a strand of DNA. You can see how this happens in the diagram below. So, transcription uses the sequence of bases in a strand of DNA to make a complementary strand of mRNA. Triplets are groups of three successive nucleotide bases in DNA. Codons are complementary groups of bases in mRNA. So let's move on to steps of transcription. Transcription takes place in three major steps: the initiation, elongation, and termination. The steps are illustrated in figure below. In initiation, it is the beginning of the transcription, obviously. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so that the enzyme can read the bases in one of the DNA strands. The enzyme is ready to make a strand of mRNA with complementary sequence of bases. Elongation simply is the addition of nucleotides to the mRNA to the messenger RNA after the initiation, after the promoter signals the DNA to unwind. So in elongation, we are adding a whole bunch of nucleotides to the mRNA strand. Now, in termination is the ending of transcription. mRNA strand is complete and it detaches from DNA. So, the steps of transcription occurs in what I said is the initiation, elongation, and termination. So in eukaryotes, the new mRNA is not ready yet for translation. At this stage, it is called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. The processing may include splicing, editing, and polyadenylation. This process modify the mRNA in various ways. Such modifications allow a single gene to be used to make more than one protein. Splicing removes entrons from mRNA, as shown in the diagram. Entrons are regions that do not code for the protein. The remaining mRNA consists only of regions called exons that do code for the protein. The ribonucleic proteins in the diagram are small proteins in the nucleus that contain RNA and are needed for the splicing process. Editing is changes of some of the nucleotides in mRNA. For example, a human protein called APOB, which helps transport of lipids in the blood, has two different forms because of editing. One form is smaller than the other because editing adds an earlier stop signal in mRNA. Now in polyadenylation, in this process, it adds a tail to the mRNA. The tail consists of a string of A's, which means it's an adenine base. It signals the end of mRNA. It is also involved in exporting mRNA from the nucleus, and it protects mRNA from enzymes that might break it down. So, the first process, which is the transcription. In transcription, we're just transfer of genes to mRNA and information, so that we have a new RNA that can be converted in protein in a translation process that we will discuss in the next video. So we have a question right here. So as a sort of review, the first question says, where does transcription takes place in eukaryotes? Go on and pause the video to see and study where this transcription takes place in a eukaryotic cell. Remember that in eukaryotic cells, transcription takes place in the nucleus, while the transcription is used happens in the ribosomes. The promoter is a region located in the A DNA, B mRNA, C tRNA, D both A and B. The correct answer is DNA. Remember that it occurs when the enzyme RNA polymerase binds to a region of gene called the promoter. The last question is the conversion of messages carried by mRNA into amino acid sequence is called A. Replication, B. DNA repair, C. Translation, D. Transcription. The correct answer of the question is letter C, which is our topic for the next video. In translation from the transcription, It is the process of conversion of mRNA-based sequences into an amino acids. That's it. I hope you've learned something. Stay safe, get